Hi everyone, I am Ankit Mishra and I have been teaching data chemical engineering from last 4 years. So in this lecture I will be discussing enzyme kinetics, that is an important topic for GATE 2025, and I am very confident about that 2 marks question will appear from this topic in the examination. So I request you all to subscribe my YouTube channel and share this lecture with your colleagues to help them in their preparation, right? So in this lecture, first I'll let you know that what sorts of thing that I'm going to discuss. That is, I'll be talking about the enzyme kinetics and these two important equation that is there in GATE syllabus, Michaelis Menten kinetics and monod model. I'll be talking, and after that I'll be talking about the enzyme inhibition, also competitive, non-competitive, and in uncompetitive inhibition. So I think all of these 4 points or 5 points are very important for GATE examination, and the questions will come from this particular topic only, right? So okay, so without wasting the time, let's just start with the topic first. So first I won't be able to, I am not going to talk about what is enzyme and what is kinetics. So directly I'll be moving towards the enzyme substrate reaction, right? Something like that. So first I'll tell you what is enzyme. So enzyme is protease biocatalyst that is required for carrying out any biochemical reaction in our body. So in our body, that just like, let's say in chemical engineering we study about the catalysis. That catalysis decreases the energy and fasten the process of the reaction. So enzyme also works as a biological catalyst, something like that. So if I'm talking about if there is a substrate and if there is an enzyme, right, and this is going to form an enzyme substrate complex having reversible reaction, and rate constant is k1 and k2, and this enzyme substrate complex, let's say es star, it will get dissociated having rate constant 3 e3 k3, and in product plus again enzyme, right? So these things are happening here. So S is the substrate, P is the enzyme, ES star is the enzyme substrate complex, and P is the product here. So if I want to write minus rate of reaction in terms of substrate, so I can write K1 concentration of S into concentration of enzyme minus obviously K2 concentration of ES star, that is enzyme substrate complex, and let's say this one is my equation first, and if I want to write rate in terms of enzyme substrate complex RES, so I can write here that K1 concentration of E and concentration of S minus K2 concentration of ES star minus K3 minus K3 and concentration of obviously ES star and concentration of S2O. So here this reaction will happen in presence of S2O something, right? That's why concentration of S2O is required here. Okay, fine. So when we talk about ET, so ET is called as the E note, that is the initial concentration of enzyme, that is total concentration of enzyme also. So the value of ET, which is equals to E naught, that can be written as obviously E plus enzyme substrate complex. Is that clear? So only enzyme is going to form enzyme and enzyme substrate complex. So from here we can calculate concentration of E, that can be written as concentration, that is E naught minus ES star. Is that clear? Something like that. Okay, so now if I want to write that R in terms of ES star, so that can be written as K1 concentration of E, and now I'll be replacing this concentration of E with this E naught minus ES. Is that clear? So let's try to understand these things. E naught minus ES star, and obviously concentration of S minus K2 concentration of ES star minus K3 concentration of ES star and concentration of H2O. So first assumption that I am going to take here is K3 and concentration of H2O, because H2O concentration will be very high, so it is constant, and I will be assuming this is a K dash. Is that clear? And then I can write RES star, that is equals to K1 concentration of E naught and concentration of S minus K1 concentration of S into concentration of ES star minus K2 concentration of ES star minus K3 dash and concentration of ES star. Is that clear? So let's try to calculate rate in terms of this RES star, that is equals to, let's try to write ES term in one side. So I can write K1 concentration of E naught concentration of S minus, if I want to take common concentration of ES star, so I can write K1 concentration of S minus K2 plus K2 and plus K3 dash. Is that clear? So since ES star is a transition complex, so if I use the steady state approximation, so rate in terms of ES, that will be equals to 0 for a steady state approximation. So that can be written as K1 concentration of E naught concentration of S minus concentration of ES enzyme substrate complex, and obviously K1 concentration of S plus K2 plus K3 dash. Okay, so if I want to find out what is concentration of ES star, that can be written as K1 concentration of E naught concentration of S divided by obviously K2 plus K3 dash plus K1 into concentration of S. Is that clear? So this is very important parameters that is used to, that will be used to calculate the Maxwell's Menten kinetics equation. Okay, now fine. So after that you can say, if I want to write a rate of reaction, so rate, because since we have seen that this equation, enzyme is going to binded with substrate, it is forming an enzyme substrate complex, and after it is producing product and E. So obviously enzyme substrate is complex is going to form, but when enzyme substrate will break, enzyme substrate complex will break, then only you can get the product. So this step is your rate determining step. So if I want to write the rate of reaction, that my R ES star, so that can be written as K3 and concentration of ES star, right? So concentration of ES star we have already calculated. So R ES star, that is equals to K3, and concentration of ES is K1 concentration of E naught enzyme initial concentration of enzyme multiplied with concentration of S divided by K2 plus K3 dash plus K1 and obviously concentration of S. Okay, so let's say if I divide this by K1 in the numerator and denominator, I can write RES star, that is equals to K3 concentration of E naught and concentration of S divided by K2 plus K3 dash divided by K1 plus concentration of S. So here I am going to define some parameters. Let's say Vmax. Vmax is maximum rate, that is K3 and concentration of E naught. So Vmax is the maximum rate for enzyme substrate reaction. Maximum rate. And if I want to define K2 plus K3 dash divided by K1, that can be called as Km, that is Maxwell's constant, right? This is called as the Maxwell's constant. Okay, so these two parameters are also very important when we talk about the enzyme substrate complex reaction. So then I can write RES star, that can be written as Vmax into concentration of S divided by Km plus concentration of S. Is that clear? And this equation is called as the Maxwell's Menten equation or kinetics, right? Now if I'm talking about this curve looks like a sigmoidal curve, right? So if I want to plot this curve like rate versus substrate concentration, let's say RES star versus substrate concentration, so you will see it will start something like that and it will go and it will follow something like that. Okay, so if I'm talking about that first point, if concentration of S is very greater than Maxwell's constant, so what will happen? Km will tending to 0. So in that case, what I can write, RES, that can be written as Vmax into concentration of S divided by concentration of S. So here you can say this will get cancelled out and that will be your Vmax. So you will be getting maximum rate, and the rate is independent of the concentration of the substrate. So at higher concentration in this particular region, you will see if concentration of S is very greater than Km, so this is called as the Vmax, that is maximum rate of the enzyme substrate reaction, and that is called as the Vmax. And here rate is independent of the concentration, so rate is not going to function of substrate concentration, right, in this particular. So that is a zero order reaction, it follows zero order kinetics first. Second point is that if concentration of substrate is very less than Km, so in that case what will happen, concentration of substrate I am going to neglect it. So then if I'll write RES star, then that can be written as Vmax into concentration of S divided by concentration of S plus Km. So here I am assuming this one is 0, so you can write Vmax divided by Km into concentration of S. So when, and this is going to follow the first order kinetics, right? So first order kinetics. So in that case, what you will see, so here what you will see, that it will follow first order kinetics, something like that. When concentration of S is very less than Km. And just try to understand one more point here. Third, that if concentration of substrate is equals to Km, what will happen? R concentration of ES star, that is equals to Vmax into concentration of S divided by concentration of S plus Km. So I can write that is Vmax into concentration of S divided by 2 into concentration of S. So this will get cancelled out, so you will see that Vmax divided by 2. So here in that case, the rate will be equal to half of its maximum value. So in that case, you will see that when Km equals to concentration of substrate, this rate will be equals to Vmax by 2, maximum rate by 2. So you can also say that the Km Michaelis constant is the concentration of substrate required to make the rate half of its initial value or maximum value. Sorry, rate will becomes half of its maximum value, right? So at that particular concentration, Km will be this one. So let's say we have three enzyme substrate these things. So let's say you have Km1 is greater than Km2 is greater than Km3. So which enzyme substrate or which substrate will be work better? Or let's say if Km is higher, then you require higher substrate concentration, right? So efficiency of enzyme will be less, right? So efficiency of enzyme will be less. So we have three enzyme, let's say. So we have, let's say, three enzyme 1, enzyme 2 and enzyme 3, and for enzyme 1 Km1 is there, for enzyme 2 Km2 is there, and for enzyme 3 Km3 is there. And if I am saying Km1 is greater than Km2 is greater than Km3, means sorry, these three substrate are there, substrate 1, substrate 2 and substrate 3. And if I am telling Km1 is greater than Km2 is greater than Km3, so if Km1 is higher, then higher substrate concentration is required, right, in order to carry out the this kind of reaction. So if Km is high, then less efficient substrate is, right? So substrate 1 will be less efficient compared to substrate 3. So Km is inversely proportional to efficiency of substrate. Is that clear? One thing. Now coming to the Vmax equation or R equation, that R that was equals to Vmax into concentration of S divided by Km plus concentration of S, right? So if I write this equation, 1 upon R that is equals to Km into concentration of S divided by Km plus concentration of S divided by Vmax into concentration of S. So I can write 1 upon R, that is equals to Km divided by Vmax into 1 upon concentration of S plus 1 upon Vmax. Okay. Okay. So here you can see that this is just a linear equation. So if I want to plot this equation something like that, 1 upon R versus 1 upon substrate concentration. So you can see this equation looks like y equals to mx plus c. So this equation will go something like that. The intercept will be 1 upon Vmax, and the slope of this line will give you the value of Km divided by Vmax. So sometimes they will give you this kind of curve. So you have to understand this curve. On y-axis this is 1 by R, and on x-axis this is 1 upon substrate concentration. And this concentration, this plot will give you that intercept of this plot gives 1 upon Vmax value, and the slope of this plot gives Km by Vmax value. Is that clear? And this equation is called as the line Weber and Burke plot, right? So based on this equation you can get all of these kind of equation unknown data also in the problem if graphical question has been asked. Okay, now one more thing is that here, if you have been given this kind of curve R versus concentration of S, and if let's say you have 3 substrates something like that, you have 3 substrate, okay, and Vmax is this one, Vmax is m. If I want to calculate Vmax by 2 here, by 2, so Vmax by 2 will give you the concentration of, let's say Km1, Maisley's constant Km2, and obviously Km3. And if this one is substrate 1, substrate 2, substrate 3, so here what we can say, Km3 is greater than Km2 is greater than Km3. And what is Km? Means concentration of substrate required to at that particular concentration of substrate the rate will becomes half of its initial value. So what you will see, for substrate 1 Km is less, means for lesser concentration of substrate you will be able to carry out, or the reaction will be very much efficient. So you can say S1 will be greater than, efficiency of substrate S1 is greater than efficiency of substrate S2 is greater than efficiency of the substrate S3. So this one is the order of efficiency of the substrate. Is that clear? I hope this is clear to all of you. So this is all about the these things, monode model kinetics, sorry, Maisley's maintained kinetics. After that I will be talking about the types of enzyme inhibition. I'll be talking a bit fast here because, so when we talk about the enzyme inhibition, that occurs when a molecule binds to an enzyme and decreases its affinity. So let's say if this one is the enzyme, so let's try to understand. If this one is the enzyme and if this one is the substrate, so this is enzyme and this is substrate, and after that this is making a enzyme substrate complex, right? But here what you will see that the enzyme and substrate work as a lock and key model. But if another molecule, when enzyme and substrate comes, it will get locked, and after that the enzyme substrate complex will form and the reaction will proceed. But if let's say if some another molecules will come, that is the inhibitor, and what will happen, this inhibitor will get binded with the enzyme also, and that will reduce the affinity of the substrate towards the enzyme. So due to this particular reason, what will happen, this decreases the activity of the enzyme about the substrate, right? So in this case you can say that enzyme inhibition occurs when a molecules binds, this molecules will be your inhibitor, binds to an enzyme and decreases its activity. That's it. So when we talk about the enzyme inhibition, so there are three different kind of enzyme inhibition. So first one is your competitive inhibition, second one is your non competitive inhibition, and third one is your uncompetitive. So these three inhibitions are there. First I'll start with the competitive inhibition. So in competitive inhibition, what you will see that the inhibitor competes with the substrate for the enzyme's activity, right? So let's say if this one is the enzyme and some inhibitor will be there and substrate will be there. So now what will happen, in order to acquire this active site, this inhibitor will be also tending towards the enzyme, and this substrate will be also tending towards this this enzyme. So here you will see there is a competition between inhibitor and substrate for enzyme active site. So this one is the enzyme's active site, let's say, and here enzyme substrate complex reactions will be happening. But here, due to this inhibitor, what will happen, both enzyme and substrate are competing with each other in order to acquire that active site, right? So I mean, if you'll see, it has a similar shape to the substrate and binds reversibly, preventing the actual substrate for binding. So here what we'll see, that substrate and obviously this inhibitor will be having a similar kind of shape with the substrate for the enzyme substrate complex formation, and due to this similar shape, what will happen, the enzyme substrate will also, I mean, this will reduce the active site for enzyme substrate complex formation. So due to this competitive inhibition, what will happen, the value of Km will increases, and if the value of Km is increasing, then I have to already discuss these things, that you require larger substrate or higher substrate concentration to carry out the exact same enzyme substrate reactions, right? Means your substrate is becoming inefficient for carry out this enzyme substrate complex reaction, and the product formation will be getting delayed. First thing. Second thing is that what is the effect on Vmax? So Vmax is not going to change. So in competitive inhibition, this is two major equation that you'll see. Km value will increase and the Vmax is not going to change. So if you see the result of this reaction here, that will be like that, Vmax into concentration of S divided by Km into 1 plus 1 upon Ki plus concentration of S. So here you will see that there is a factor more than 1. This value will be more than 1. It is getting multiplied with the Km value, and that's why it is increasing the value of Maxley's content Km. Okay. Okay, and Ki is called as the inhibition constant. Ki is called as the inhibition constant. That is very, very important, that you have to understand. Now, second thing that I'm talking here is the non-competitive inhibition, right? So okay, So here what you will see that inhibitor binds to both the enzyme and the enzyme substrate complex. Right. So due to this particular reason, it will decrease the value of Vmax, but the value of Km no change. It is not going to change the value of Km, but the maximum rate is getting decreased, right? So the equation that you will see that R, that is equals to Vmax into concentration of S divided by Km plus concentration of S into 1 plus 1 upon Ki. Is that clear? Okay. Now the third and the last inhibition, that is your uncompetitive inhibition. Here what you will see that inhibitor binds only to the enzyme substrate complex. So these kind of questions can be also asked, that where inhibitor will bind. So inhibitor is going to bind with the enzyme substrate complex only, and it will decrease the value of Vmax and it will also decrease the value of Km. So both of these value will get decreased. That determines the kinetics of the enzyme substrate reaction. So if I want to write the reactions here, that can be written as Vmax into S divided by Km plus concentration of S multiplied with 1 plus 1 divided by Ki. Is that clear? So this one is the equation. So we have seen three kind of inhibition. That is, first competitive inhibition, where inhibitor was binding with the enzyme only, and it was decreasing the active sites available for substrate and reducing the value of Km. So in that case, you will see that more substrate concentration was required to carry out the same reaction. So that was decreasing the efficiency of the substrate. In the second case that we have seen non-competitive inhibition, where inhibitor were binding with the enzyme as well as enzyme substrate complex both. So ultimately it was reducing the value of Vmax, that is maximum rate. And the third case that we have seen is the uncompetitive inhibition, where enzyme binds with only enzyme substrate complex, and it was ultimately decreasing both Vmax and the value of Km. Okay, so if I am talking about this thing, if let's say if we make a plot something like that, Lineweaver work a plot something like that. So in one case Km was decreasing, one case Vmax was decreasing, and one case, in another case, this was first case, this was second case, and this is third case for the uncompetitive inhibition, where Km was decreasing and Vmax was decreasing. Right. So if I plot 1 upon R versus 1 upon capital S, so a plot was something like that, you can see. Okay, so this was the plot. So let's say, okay, and slope was Km divided by Vmax, right? Slope was Km divided by Vmax. Okay, so if Vmax will increase, slope will decrease, and 1 by Vmax will also decrease. So 1 will go something like that. And in another case, Km was constant and Vmax was decreasing and Km was decreasing. Okay, Km was constant, Vmax was decreasing. So, okay, so these three cases are there. So here you can see that slope is Km divided by Vmax, and here slope is more, here slope is less. Is that clear? So first graph is for competitive inhibition. If Vmax will decrease, slope will increase. So sorry, this is for non-competitive inhibition. This is for competitive inhibition, and this is for uncompetitive inhibition. Is that clear? So you can make this kind of plot also, and from where you can understand all of these kinetics for competitive, non-competitive and in-competitive inhibition. That is very, very important curve. And also sometimes you can plot this kind of curve like that let's say in sigmoidal form, if this curve is looking like that, and if this is V, that is rate, if this is R versus concentration of S, and if you want to plot this curve, so you can have a curve like that. Okay, and again you can have a curve like that, and again you can have a curve like that, right? So if you want to understand this curve, so this will be your competitive inhibition, and this one is your uncompetitive inhibition, and this is your non. So from the Vmax value, this one is your Vmax. Is that clear? And again you can calculate the value of Vmax by 2 from here. Let's say this is Km and this is your Vmax by 2. You can check whether this one is Km1, this one is Km2, and this one is Km3. So for which Km is decreasing, and after that you can calculate all of this. Is that clear? Okay. If it is clear, then I'll be moving towards the next discussion here. And this is very, very important. I think GATE 2023 asked questions from here that if MM kinetics is happening in plug flow reactor, if it is happening in plug flow reactor, then what we can see, that rate we know that is equals to Vmax into concentration of S divided by Km plus concentration of S. So here we can say dCs divided by dt, that is equals to, or we can say ds by dt is equals to Vmax into S divided by Km plus S. S is the substrate concentration. Is that clear? So from here we can say ds by dt and Km plus S, that is equals to Vmax into S dt. So from here we can say Km by S ds plus S ds is equals to Vmax in Vmax dt. Is that clear? So if I'll take common Km and integral, it will be going from ds to S plus S ds integral, that is equals to Vmax dt. So it will go from S naught to S, let's say S naught to S. So I can write Km into ln S divided by S naught plus S minus S naught, that is equals to Vmax into T. So you can calculate the time required to decrease the concentration of substrate from S naught to S. So if in S naught is the initial substrate concentration and S is the concentration of substrate at any time t, and Vmax is obviously maximum rate and Km is the Michaelis constant. So from this formula you can say that time is equals to 1 upon obviously Vmax, 1 upon Vmax into Km ln S divided by S naught plus S minus S naught. And all of this value will be given to you, and you can calculate the time required to decrease the concentration of a substrate in plug flow reactor from S to S naught, where Km is the Michaelis constant, right? Now, second is for if this reaction is happening for CSTR. Let's say you are having a CSTR something like that, right? So CSTR is continuously in, continuously out. This is the volumetric flow rate of the reactant, and tau residence time is given to you, and obviously concentration of reactant S naught is initial concentration and S is the final concentration. S is the concentration is the inside the CSTR, and if I am assuming V is the volume of the CSTR, so what is tau? Tau can be written as S naught minus S divided by R. And what is S naught minus S, and what is R? R is your Vmax into S divided by Km plus S. So finally the space time can be calculated as, let's say, S naught minus S into Km plus S and divided by Vmax into S. So while using this equation, you can calculate the time required for space time in CSTR, where S is the concentration of substrate at the exit of CSTR, and S naught is the concentration of substrate at inlet of CSTR, and V naught is the volumetric flow rate. So these kind of things that can be asked in your examination, so you have to be prepared with that thing. Okay, so this is all about that, how this kinetics is happening in plug flow reactor and CSTR, and how you can calculate the space time for CSTR and time required for decrease the concentration of substrate from S naught to S in a plug flow reactor. Okay, after that I'll be moving towards the last concept of this discussion, that is your monode model. And monode model is also very important kinetics that can be asked in your GATE examination. So let's understand the monode model. So first we need to understand where we can use the monode model. So the monode model describes the microbial growth kinetics in response to the substrate concentration, right? So this is also analogous to the Michaelis-Menten kinetics that is used for the enzyme kinetics, but this is for the microbial growth. This is called as the microbial growth kinetics, and the equation for the monode model looks like mu equals to mu max into S divided by again Ks plus S. So it is exactly similar to the kinetics, but here the parameters that is being used in this equation is different, where mu is called as the specific growth rate of micro organism, mu max is maximum growth rate, and S is the substrate concentration, and obviously Ks is the half saturation constant, which is equals to mu max by 2, something like that. So with the help of monode model also, you can see that at low substrate concentration, if you are going to increase the substrate, growth rate will increase, but at higher concentration it is going to get saturated. So how much extra amount of substrate that you are using, substrate is not going to affect these things, right? So again, the graph of this monode model also, it will look like that we have seen that sigmoidal curve in. So it will go something like that, and it will get saturated at higher substrate concentration. So here S will be very greater than Ks, and here you will see this will also follow first order reaction, and in this case S is very less than Ks, and S will be equals to Ks at this particular point, and that will be mu max, and here you will see mu max divided by 2 at when substrate concentration becomes equals to the saturation constant value. Is that clear? Okay, so this was all about the enzyme kinetics, enzyme inhibition, and microbial growth kinetics, that is monomer model kinetics, something like that. So all of these things becomes very important. I have already discussed all of these things. I hope you will get a question in the GATE examination in 2025, and you will be able to solve the question from here on. Okay, thank you all. See you in the next lecture.